Once again, good afternoon, everybody. Thank you for joining us for this online talk. I hope that it will be full of insights and full of lively discussions that follows after. Perhaps a little bit of about myself. My name is Boedi. I was born in a small city in central Java, Indonesia, a city called Solo. And I, together with my siblings, migrated to Singapore at a very young age due to certain socio-political situations that was happening in my country at the time. And much of my artistic practice revolves around my childhood memories of being away from home, being away from my parents at the time, and also has a lot to do with my experiences as I navigated my way through this very foreign place, this big city called Singapore. This trilogy of projects that I will be talking about today has a very direct relationship with these childhood experiences. I will start right from the beginning, the genesis of this trilogy of projects. That was something I conceived in 2019, the year before the global pandemic. And the project began as a work titled A Tree Talks, A Tree Walks, Wu Tong Yu, Pu Ti Jing. It started at an artist residency in Singapore, a place called Temangong Artist in Residence. It was a very interesting place because it was an old colonial style building, and it was located at the foot of a hill that faced the south. It faced the Singapore Straits and the Java Sea towards my hometown, I guess, if you can say that. And these are some photographs of the exhibition of the works that were developed during the residency, A Tree Talks, A Tree Walks. It was part of a two-artist exhibition. And the exhibition also had a talk that allowed the artist involved to explain about the projects that we developed during the residency. So it was an interesting beginning for this project because when we were invited, when the other artists and myself were invited to this artist residency, part of the premise of that residency had to do with a site-specific approach or response to the residency. And one of the most striking things that left a deep impression upon me were the many Chinese calligraphic pieces that were hanging on the walls of this residency. The photographs that are right now here on this slide is an exhibition of a pioneering artist in Singapore named Parasol, where his calligraphic works were presented. But on top of the works by Parasol, there were also many works that belonged to the collection of the owner of this residency. And these calligraphic pieces in his collection were from the different dynasties of in China, and I was very fascinated by the presence of writing as an art form in this place. My paternal grandfather, his name was Wu Tong, he was named after the Chinese parasol tree, had lived together with my family in the last few years of his life. And I got to know, I think, in my teenage years or so, that he had left behind a journal. And in the journal, he had penned down his life experiences as, I guess, through the framework of a Chinese minority living in Indonesia at the time. He detailed his family life, the thoughts that he had about certain family members, and so forth and so forth. And it was a precious journal for me because I did not have many memories of my grandfather. He lived, I think, around 2 or 3 years together with my family, and after which he passed on from an illness. But throughout that 2 or 3 years, I had a great time interacting with him, and I have very fond memories that remain very vivid, even now as an adult. And so I had always wanted to somehow use the journal that he left behind as a material for one of my art. And when the artist residency at Temanggung came about, I felt that it was the right time to relook at this journal because my grandfather's writings became a very potent raw material in the context of responding to my fascination towards the presence of the Chinese calligraphic pieces at Temanggung. And also, I think at the same time, I am very curious about the kind of connections that I can draw and imagine through the words and memories of my late grandfather. About 2 or 3 years before the residency, I had came across this very interesting article in the New York Times newspaper, and it led me to think about what can somebody inherit through one's genetics or epigenetics in this case. The article very briefly talked about how trauma or suffering can possibly be passed down through chemical markers that are found in our DNA. And these chemical markers would then have an impact in the way DNA is read and translated into proteins in our body. And hence, this idea of being able to inherit the experiences or the memories of our ancestors fascinated me. And I wanted to use this project as a way to develop my thinking around this interest. Now, this project wouldn't have been possible if I hadn't had a chance to meet with Associate Professor Dr. Eric Yap, who is a medical geneticist at the Lee Kong Chian School of Medicine at NTU Singapore, who had kindly and so generously helped me in so many ways in this project. So I think we will later talk a little bit more about our collaboration and the experience that both of us derived from that. So how do we combine the idea of ancestral experiences and memories with my fascination with writing or writing systems, the presence of words and letters with the DNA? One of the big gestures in the artwork, A Tree Talks, A Tree Walks, had to do with first synthesizing these disparate elements together. And the idea here is to come up with a hybrid DNA, a chimera of person, plant, and text or poetry. Okay. And this is how we did the hybrid DNA. If you are to look at these letters on the screen, each unit being formed by 3 letters. These are the basic units of the DNA language called codons. And the hybrid DNA, as indicated by the 3 colors, was made up by 3 sources. The first being a part of my Y-chromosome, which was obtained through my saliva, or the cells that was harvested through that. And there was a particular part of that Y-chromosome that was used in this DNA marker called the YSTR marker. I will let Dr. Eric elaborate about why this part of the chromosome was chosen. The green part of the hybrid DNA was actually taken from the chloroplast sequence of a Chinese parasol tree. I mentioned that my grandfather's name was Wu Tong, and so we felt that taking a part of the tree would be meaningful. Something interesting to say here was that Wu Tong wasn't my grandfather's original name. His actual name was Cheng Jia, and he was adopted as a youth when he was still in Fujian, China. And he had to change his name and adopt a new surname in order to be able to migrate to Indonesia as a family member of his new family. Hence, the name Wu Tong was, in my mind, very significant as a symbol or as an expression of his journey to the south or towards Nanyang. And the third and last part of the hybrid DNA was an encoded text of the title of the artwork, except that the title was written as a transliterated form of the Chinese Hanyu pinyin of the English title. I will explain a little bit more about this in yeah. So again, just to talk a little bit about the basic syntax of the DNA. It is made up by 4 letters, A, T, C, and G. And each unit would draw from these 4 letters only 3 at a time. And they are called triplets or codons. So how do we encode the artwork's title in the language of the DNA? First, I would have to speak a little bit about traditional Javanese language that I learned before I migrated to Singapore. This was something that I learned in school. The language or the script is called hanacaraka, and it is an ancient Javanese script. The modern version that is still being used right now, if you ever go to Solo City right now, you would be able to see this script used as road signs or even as a part of the signages in front of retail shops and so forth. But the script had originally came from India, first as a Pallava script before it was developed in Java into the Kawi script, and before in the around the 15th century, it was further developed into what we know today as this modern Javanese script. And the hanacaraka is a very interesting script for me because, first of all, it embedded within this script is a very powerful story of a death duel between two subjects of a king who had a misunderstanding. Both of them had misunderstood, I suppose, the instructions given to them by their kings. And in order to follow their respective instructions, they fought and both died as a result. And this catastrophic outcome or remains of the clash of two great powers is a narrative that I think about a lot in my personal narrative. Of course, it directly is a metaphor of the political transition around the 1960s, in the mid 1960s in Indonesia, that had directly impacted the policies towards ethnic minorities in Indonesia, but also to the bipolar geopolitical situation of the time, the Cold War, that enveloped and framed that same political transition that I talked about. So hanacaraka not only reminds me of my place of birth and my childhood experiences in Solo, my Javanese heritage and memories, but the meaning or the story embedded in it is something that resonates a lot with my personal narratives and in how I think about the histories that influenced and framed this narrative. The hanacaraka script is it belongs to a category of languages called the Abugida script category, and by that it means that each letter is represents a consonant, and through the use of diacritics or additional marks added to it, one would then read it differently. Meaning the vowels are indicated through these different diacritic marks. And this emphasis on the consonants gave rise to the idea of combining or mapping across the 20 amino acids that are coded by the 64 different possible codons in our DNA, with the 20 hanacaraka letters that we saw just now. And this became an encoding key to encode any text or words that are written using hanacaraka phonetic spelling into the language of DNA or the codons. The hybrid DNA, the material of it, was then used in a site-specific installation. This is a photo of how the installation looked like at the Mangong Artist in Residence. We can't see it from these photographs, but the cross poles of the flags behind the trees was actually the Java Sea. So these flagpoles were addressing the orientation towards Java. And below the flagpoles is this mound of soil. This is a close-up image of the mound of soil that was marked using a mixture of ink and the hybrid DNA. So across the exhibition period, we invited members of the audience to use this mixture of ink and soil to mark this installation. On the photo on the left, you would be able to see at a distance down the slope a hole from which the soil was taken from. A close-up of that you can see on the image on the right. And there was this idea of returning this soil that had been taken up from the land back into where it was taken from. And this is what we ended up doing at the end of the exhibition. We took the soil that had been marked with the hybrid DNA and placed it back into the hole from which it came from. And it was really a gesture of rooting oneself into a new place. Or in Chinese, there's a Chengyu that says, 落地生根. Yeah. And these are images of the exhibition inside the colonial house at the residency. There are parts of that soil installation was presented in the vitrine that you are seeing right now in the image. The diagram below on the floor serves as a compass with the corners of the square pointing towards the different orientation. And these are more installation images at the artist in residence. I would like to elaborate a little bit more into two works that were presented at the residency at the Mangong. These are visual writings that were developed for the exhibition. And the first work, the one on the left just now, this is a detailed view of it, is an encoding of a poem originally written in Sundanese, one of the languages of ancient Java. And this poem, interestingly, captured all the landmarks across the Java island. The story had it such that a prince from Sunda, a kingdom in West Java at the time, traveled by foot across the island, and as he walked, he would note down the different landmarks that he came across. And the result was a poem that was, in a way, also a map of the entire island of Java. And hence, I took a fragment of that poem and encoded it in this visual writing system for this work. So the image that you saw just now was actually made up by a sequence of hexagrams. Once again, the idea here is to think of the DNA language as a way to connect myself to certain cultural sources that are meaningful to me. And this is just to refresh. The 64 codons on the right column were mapped to the 64 hexagrams that are found in the I Ching. And because of the previous mapping of the amino acids that the 64 codons had mapped for with the 20 hanacaraka letters, I was then able to connect through the DNA language the 64 hexagrams in the I Ching with the 20 Javanese or hanacaraka letters. And it was very meaningful for me because through this exercise, I was able to bridge two cultures or myths of cultures that I couldn't quite reconcile growing up as a child in Singapore, as a foreigner in Singapore. So it was something that was particularly meaningful for me in thinking about my cultural origins and my cultural identity today. The form of the work, of the visual writing, also drew from the continuous script format that are found both in the hanacaraka writing and the DNA sequencing. So I developed two works along this series. The red colored piece just now was of a Sundanese poem, and the other work, the one that was colored in black, was an encoding of my grandfather's journal. And the process that I took for this second work, titled Nanyang, had first to do with transliterating my grandfather's journal through my father's voice. So I'm just going to play for a while my father's reading of the journal.
以1911年11月降生一个男儿乔木，母亲因我的男孙而惋惜不已。暂时家庭情况安静。
So the reason why I had asked my father to read my grandfather's journal was because I think that there is a kind of cultural signature or characteristics that were embedded in the way he read these words. And I wanted to capture that cultural signature or characteristics, if you like, and bring it into my encoding. So this is how I did it. I listened to how my father read the text or read the journal, and based on his pronunciation, I then wrote them down using the phonetic spelling of the hanacaraka script. And from here, using the visual writing coding system, the hexagrams were then used according to that visual writing system. So this is a close-up view of the work Nanyang. The white parts, by the way, are the where there was where the line breaks and punctuations and so forth took place. So that wrapped up the first project in this trilogy, the work that was later further developed into the second project, 3 rings, 3 sings, Shuling. Where the first project thought about genetics or the writings found in our body. The second project contemplated the area of epigenetics, or the kind of activities of reading what had been written in our DNA, and the activities or the factors that would impact this act of reading and the outcome from that reading. And in this work, I tried to imagine that the memories and experiences we inherit from our ancestors, how would they be if they are experienced in the form of images and sounds? Hence, the idea of using the hybrid DNA as music score came about because of that question. I was at the time also reading a little bit about Brian Eno, a UK-based musician who was rather influential in shaping the discourse around generative music, where the idea of an ever-changing sound can emerge from a fixed system. And I am drawn to that idea of generative music because I think it suggests to me a kind of non-determinant kind of reading of a source text that is fixed, and it resonates with the way I imagine epigenetics, that there are variables present in the act of reading, and which would then influence the outcome, and not necessarily having the original text altered in any way. And this idea of using the hybrid DNA as a music score was also influenced with how the hanacaraka script in the Javanese courts were usually performed. It was either read out, or it might sometimes be performed as a song. Hence, the idea here is to create a kind of a music video or sound video that can play almost endlessly and with a constant kind of variance as it plays. So this is the video that I made. I'm just going to play a little bit of it. Oops. The letters that are being highlighted are that part of the hybrid DNA that was read by the algorithm at any moment. the sounds that you are listening to now, they are gamelan sounds that were played in reverse. These gamelan sounds came from a library that I chanced upon some years back. They had been put up online as a free resource by sound recording specialists who had access to a 200-year-old gamelan in a museum in the Netherlands called Museum Nusantara. And perchance this 200-year-old gamelan had originally been acquired by the museum from the palace in Solo, which was a very interesting serendipitous coincidence. The images in this video were created by rephotographing from an original set of photographs that I took about 9 years ago when I visited the village Hong Tai Chun near Xiamen, China, the place where my grandfather was born. And for this video, I had used a lens that I had inverted to create this kind of amorphous, blurry imageries, which in my mind, I guess in the imaginings of my mind, how I would recall images of the past. The work, Three Rings, Three Things, was developed during my artist residency at NTU CCA, which was also the place where I had met with Kuen, and the place that where we discussed the possibility of my participation into the bioart project. And it was a very interesting time for me to be able to develop this generative video work using the DNA as a music score. These are images of how the work were presented during the Studio Open in January this year. And it was through this presentation that I thought about the possibility of making a third work that would complete the trilogy of my research into DNA and ancestral memories. These are images of the soil fragments from the first work that were also presented at the Studio Open. And another visual writing that I did during the residency at NTU CCA, this time encoding a poem that I had written for a performance about 2 years back. So we are coming to the third work, which is a work that I am still developing at this moment, Three Heaves, Three Leaves, Yao Ye. It is a work that began with an image. This was a photograph, a long exposure photograph taken of the video. Hence, it is a still image from a moving image. And I was thinking about the fluid nature of ancestral imageries for the diasporic community, how the sense of one's roots and beginnings or origins can be very mediated, as well, particularly so for in my own experience. And I wanted to develop an ancestral book or zupu or book of genealogy that drives not from a complete understanding of where one comes from, but that begins the process through understanding or through looking at these abstract imageries. And this is something that, of course, is still being developed as of now. But what I want to do and hope to do is to use this work to imagine the body as a living, as a changing book, as an archive that is living, that accretes information over time. And whether I can use a biomolecular method or the language of DNA to encode and store these images, videos, sounds even, and so forth, and into a work that can be that can be accessed digitally through some technology. So it is still rather vague and rather open at this moment, but this is something that I am working on right now. And hopefully it will also give me a chance to collaborate with existing technologies or experimental technologies out there to allow me to turn this work into a reality. And these works will be presented as part of a group show at FACT Liverpool, curated by Annie Kuen, with the third work being presented as a kind of work in progress. And so thank you very much for listening to my presentation. Thank you.
Thank you, Boedi. Oh my God, it's my, okay. Now we'll welcome project curator Kuen. And he will give a short conversation with Professor Yap and Boedi. So let's welcome Kuen. You can all see him now.
Thank you very much, Tiffany. And of course, Boedi, for this very stimulating afternoon sharing. My head is very much swimming with the complex ideas that you have presented to us. I should always remember, like you said, the day we met in January, just earlier this year. And then when I chanced upon your project, your trilogy project, I felt like, wow, I've struck a lottery. Right? That's finally in Singapore. I have located an artist who has very explicitly set out to deal with the bio and to seek different kinds of representation through the bio. In this dialogue that you have created, I think is even much more complex. You have sought to look into DNA and then also try to amplify them, the DNA language, into interfacing it with nature, with natural materials such as tree and soil, and also with the private and the public and the collective, you know, in this way, and also with visual and also linguistic systems. Yeah. So how you have compounded and hybridized these various languages and their own systems of organization to develop what you have also said to be like an endless, you know, a kind of endless unfolding of possibilities. And I think this is really, really exciting. And I'm sure all of us have a lot of questions. And I would encourage everyone at this point to type, to kind of think through your question and type it out as clearly as you can on the chat room, so that later we can, you know, flag them out. But at this point, I should also turn our attention to Dr. Eric Yap, who is, of course, a very important protagonist in this project, in actually kind of like, you know, enabling Boedi as an artist to enter into the field of science, right? And Dr. Eric, of course, is a veteran, having worked with very important organizations in Singapore, and being quite a very unique individual, because he's not only a geneticist, he's also a trained medical doctor. So at this moment, I would like to invite Dr. Eric to join us to share your perspective on maybe also a bit about yourself, right? Because surely as a scientist working in the labs, then suddenly one day an artist pops into your presence and asks, hey, you know, would you join me in a collaboration? I'm sure this is quite rare, and especially in the context of Singapore, I haven't come across so many kinds of such exchanges. So Dr. Eric, could you just share with us a bit about your background, how maybe also how you are interested in the arts, and how this interest and this chance encounter with Boedi has led you into this collaboration?
Well, thank you very much, Kuen, and thank you so much to the organizers for setting this very unique event up. The brokering of relationships is actually quite a magical thing, and you might imagine artists in artist colonies, scientists in scientific labs, and the twain shall never meet. As a matter of fact, the person who brought us together, I think might be in the audience. He goes by the name of, interesting name of Soto Kambing. And Mr. Vincent, you might want to reveal yourself if it's convenient. But it's from this, yes, hi, Vincent. Vincent is a mutual friend. And he kind of introduced us, and over coffee and over a chat, we kind of, I guess, you know, kind of without being too romantic, or we kind of it was maybe, and to declare we both have wives, we're both happily married, but it was kind of love at first sight. There was a little bit of effort to have to learn languages. And I think this afternoon's topic is all about language. And so perhaps I'd frame this in terms of the language of science, what I, a medical scientist, do, lots of jargon, lots of difficult concepts, and perhaps on the other camp, the language of the arts, flowery, vague, and therefore very inspiring, challenging. These two languages actually have come together today. And it's something like the Rosetta Stone. You've heard of this, the translation between languages. Maybe I will come to that in a bit later. But here is the thought. we actually, each of us are quite normal people. We have families, we have the usual financial anxieties and health anxieties. And yet, in the sophistication of human society, we have evolved into rather differing people and almost in worlds of our own. But that hasn't been always the case. And I recall as a young student being inspired by a historical scientist who was as much an artist, the Renaissance man, Leonardo da Vinci, as much an anatomist. He dissected animals, horses, humans, and he of course drew great art, and of course also invented lots of things, an engineer at heart. In the Renaissance period, there was this flowering of culture, flourishing of thought. Partly fueled perhaps by the invention of the printed word, the book, but also I think by a historical cultural scenario where there was sufficient wealth, there were patrons. Today we still have, largely speaking, sufficient wealth. We still have the patronage of governments. But we seem to have lost this age of the Renaissance age. We have lots of stove piping. We have lots of bubbles. And as a child, I didn't have. I had an exposure to a broad range of things. In fact, my parents brought me up to learn to be an amateur violinist, and my mum, I think, who's in the audience actually, played the piano, still does play the piano. But we were schooled very much in the sciences, and the science was not sectorized as it is today. So whether you're an engineer, whether you're a biologist, whether you're a physicist, whether you're a chemist, today it has evolved to super specialization. But that was something always foreign to me. So I started off life as a medical doctor, went to medical school, had the chance to operate on a few unfortunate patients. They didn't become casualties, thankfully. But eventually I had a chance to go to Oxford to see a different way of life, a more historical perspective, much more aged, where time slows down, and to see science in its philosophical aspect of science, and to appreciate the history of science. But as life would have it, I came back to Singapore and spent the next 20 years of my life as a public servant, as a scientist, working for the government. And that's where I encountered the engineers. And over time, that has brought me to my current position as an educationist, as a researcher in academia. I think it's this varied perspectives, meeting different people through life, that has helped to break down those borders, those barriers, and to actually be able to translate between, at least for myself, within the engineering and medical and biological sciences. And therefore, stepping to talk to Boedi hasn't been that far of a jump. It's just a little extension. And by the way, I must say that Boedi can almost teach biology. I think his explanations earlier on have been spot on accurate. And so I think that testifies to the varied backgrounds, and I'm sure the talent as well, to be able to translate between these fields. And that's really, I think, the fun of it all, that we are really talking the same concepts. We may have started with different languages, but eventually, as we learn each other's language of science and of the arts, we find that yeah, we share a lot of the same ideas. Back to you, Kuen.
Thank you, Dr. Eric Yap. Okay, so I'm curious if Dr. Yap, if you were to revisit this project, this collaboration and dialogue with Boedi, would you be able to locate for us, apart from this common and this ease of approaching each other on an individual to individual level, but let's say, speaking as a scientist, if you were to revisit what Boedi has done, and through the process of working with him, what would be some of your own reflections over this period of time? Is there something you would change, or would you have proposed to him to think differently, or would you even challenge this project? Although he's of course the main author of the artwork.
Well, that's a difficult question to answer, Kuen, especially when the antagonist is in the audience looking at me. But I'm just joking.
Or what queries would you still have about artists approaching science? Let's say.
Yeah, back to your original question, would we have done things differently? I'd say an artist would have repainted his artwork differently. The second time would never be the same as the first time. And it may surprise you to hear that, well, I would say scientists would do exactly the same thing. Much as you would think of the accuracy and reproducibility of science, the rigor and permanence of science. But the truth is that science is very fluid. Science will change. And the way we would do things would change immensely as well. So let me give you an specific example. So this work we did in 2019, of course pre-COVID. The forming of this chimera, this hybrid DNA, was done by a method Boedi didn't mention, but it's a method that at that time, I would say perhaps maybe a fraction of you might know, 10% of you might know if we had held this forum 2 years ago. But today, I think without exception, each one of us knows the method. It's called PCR. And thanks to the pandemic, we all are much more familiar with a lot of the techniques, with the diagnostics, about public health and about viruses, so much more. The method we used is actually won the Nobel Prize in 1993. So it's a cutting edge method, PCR. And what it does, for those of you who can't remember, it's basically a molecular biologist photocopying tool. It generates billions of copies of an original DNA. And this is what is used to do COVID testing today. But there's another function of PCR that you may not know. And that is it's able to stitch together and to therefore create novel pieces of DNA. And so this is what the technique that we use to join these pieces of DNA from a tree, from a artificial text, as well as from Boedi's own chromosome Y. Now, if we were to do it today, we might use a very different method. And that's the field of, because technology has changed even in the last few years. And today we can actually synthesize or manufacture this DNA straight on machines. And this is the field of synthetic biology, which is able to generate not just DNA and not just short pieces of DNA like what we did, but longer pieces, whole genes, and in fact, whole chromosomes and genomes. And that's an exciting area to go into, because we can now use the ideas that we were experimenting with in a very limited way, with just one human gene, one plant gene, and a short text. We might now be able to do that a million times, in a million times larger in scope. That means a million bases, a million letters. So yes, that's something on the horizon, and that's something we would do rather differently.
Okay, cool. So it is basically the access to technologies and how technologies have progressed to enable us to, we mere human beings or non-scientists, to really be able to access and utilize them, no? In that way. So does that mean, Boedi, that you don't need Dr. Eric anymore from now on, since you know some of these magic tools?
No, I think Dr. Eric is a very critical participant and enabler of this series of works. Yeah, he's just somebody who is very humble and very giving and very generous with his words.
Cool. So now I think we have to open up the floor to some of these questions that have come in. Actually, we haven't received new questions so far, but there was a list of questions that some of you had sent in before. And I think because there are quite a number of them, we can't deal with we can't deal with all of them. So I would like to pick maybe those that seem more interesting and might provoke, right, some of these complexities that we face in meeting art and science. So one goes, what challenges, Boedi, or not, do you imagine there regarding translation and accessibility for those outside of the Asian diasporic communities? Yeah, with less understanding of the visual language systems and epistemologies of tradition and lineage.
I think this a project like this, something as personal as this, has to very much start from a kind of hyper locality. We are talking about DNA materials. We are talking about personal stories. This is really as personal, as individual as you can get, and in the sense as encoded as one can get. So I think in order for it to be meaningful and real, that kind of individuality and the encoding of that is necessary. So it must not, in my opinion, start from a consideration of the most efficient way of communicating to as large a number of people as possible. However, having said that, the use of DNA is, I think, an enabler for anybody to access the encodings and that has taken place in this work, because the DNA language is something that is embodied in every human being. Hence, it is a kind of a universal interface, or a universal material and experience, if you like, that everybody can relate with. I mean, especially so now during the pandemic, we talk about mRNA vaccines and so forth. All these terms of the molecular biology has surfaced so much into the mainstream media. And I think that, in my opinion, provides a kind of access way into the more personal, the more coded meanings that are embedded in this work. So while the so I think there's a presence of both a common language and a personal language in this work that would allow for access at one end, and the possibility to articulate in a very personal and coded form meanings on the other end.
And how do you think? That's what is quite interesting to me. I mean, like when I encountered your project, I was very drawn to how you were not merely taking this as a personal, hermetic journey into resolving some of your traumas that you have spoken of, right? Of displacement, of relocation, of the larger diasporic trauma, so to speak, but that you actually then manage through the prism of the DNA and ways of re-encoding it, so that you are also kind of like really kind of making not things reductive, but to actually complexify, and then to amplify the representations into visual drawing, into music, and into new forms of writing, so to speak. And actually, in your coming last part of your trilogy, you are even talking about a kind of living embodiment, right? A kind of like an embodiment that is constantly reinventing or renewing, never being in this stasis, right? So I think so the points of access, right? The emotional, especially, I mean, a lot of people still talk about the emotional as the anchor to perceiving artwork. I think on both the ends of the formal as well as the affective, you open up, you open up the landscape and different forms of representation in order that we are able to plug, right? To plug in where we can. Yeah. And what about you, Dr. Eric? When you are when, of course, after working with Boedi, right, and then you entered the exhibition, and then you encountered the artwork, right, its interpretation. What were some of your own revelations?
Right.
Or how were you able to access, if we want to tackle this question of communication, right, and this point of entry, how did it work for you?
Maybe I can take off from a thought that was discussed, and that thought was that of geography, of migration, of one's past, and one's language. And let me see if I can share the current screen with you. Right. I wonder whether you can see two maps on your screen.
Yep.
Yeah.
Yes, and so this relates to our past and where we come from and the locality that we are talking about. We are very much a function of where we are located, and it's true that we have all these. We have our entire past encoded in our genes, and of course, each of us carry a different version of these 3 billion nucleotides, 3 billion letters of our human genome. But what's been shown is actually that the genes that we carry actually tie in very closely with the languages that we see distributed. And these are two pictures of East Asia. On the left is actually a map showing the different linguistic groups as defined by the linguists, and the map actually shows that they're actually very closely related to the genetic map. You'll see that this branching tree is actually a Y chromosome tree. That is to say that the Y chromosome similarities actually map very closely to the linguistic similarities. And this is of course of East Asia, Southeast Asia in particular. And when one maps this to age, one can account for this by the migratory patterns. So the languages, the similarity between, say, the native languages in Philippines, in Malaysia, and the native language in the Taiwanese ethnic minorities, the relationship actually maps according to the migratory patterns. Yeah, and so do the genes. And I think this kind of and this pattern actually has been shown at a micro level. If one looks at just within a sub-region, or in fact in other parts of the world as well, in Europe, for instance, this pattern has been very clearly shown. It almost tells us that the mother tongue, maps very closely to either the mother's genes or, in this case, the father's genes, the Y chromosome. And perhaps at this stage, I just want to answer a query, if I may, on the chat. Let me just see. I saw a query on the chat from Ku Kwang Yi asking why the Y chromosome. Yeah, and I'll just say here that the Y chromosome is carried only from father to son. Now, of course, females don't have the Y chromosome, and maybe they're all the better for it. The Y chromosome is actually a rather small, miserable piece of DNA, which some evolutionists forecast that in several million years' time, it will shrink into nothing. And so the Y chromosome is certainly on the decline. You might argue like some parts of the world today. Thankfully, it's going to be on a very long scale of many millions of years. But anyway, that's a chromosome Y. It's carried only by males. And that makes the genetics somewhat simpler because it only gets carried from father to son, and we don't have access to Boedi's grandfather's DNA, but we do actually through Boedi, because the chromosome Y that Boedi has now is exactly the same as that of his father and of his father's father and of his father's father's father, and so on. So it's transmitted along the paternal line. There's also a mother's version of this called the mitochondrial DNA, very much similar, transmitted only on the mother's line. But these are just a very small part of the whole genome. And most of our DNA, most of our genetics is actually equally inherited from our father and from our mother. So before we well, before we start casting blame or too much on either one of our parents, most likely it's from both. Thanks.
Cool.
I would like to add to what Dr. Eric had just said. I think the use of the Y chromosome was quite meaningful for me because it allowed me to connect very directly to the land of where my paternal grandfather was born in, of which images that I took started to be brought back into the video work as well. Hence, going back to the visual writing systems, where I encoded both the map of Java and also, in a sense, my grandfather's journey to Nanyang. My main point of interest is to draw linkages through the DNA to the two cultures that are, to me, for a very long time, came across to be quite irreconcilable. And that was the primary motivation of connecting through, I guess, this paternal line towards my grandfather, and on one side, and also to the place of my birth in Solo at the other end. Yes. So it's I think it's a more cultural, geographical kind of a consideration that expresses itself through the DNA. Yes.
Cool. Okay, so we have had quite a bit of sharing today. It's arriving at nearly 4:30. I think if there is anything, of a farewell, bit of thing that you want to say, Dr. Eric, would you like to share, or Boedi? Yeah, before we, let's say, wrap up today's session.
Dr. Eric?
I was going to allow you to go first.
Okay. I'll go first. Thank you, Kuen and the team at this BioArt Southeast Asia Taiwan for giving me this platform to talk about this project. I think this is the first time that I talk quite in depth about the inner kind of processes of these works, and I think I had inadvertently taken up quite a fair bit of today's time. So sorry for that. And thank you, Dr. Eric, for being so generous with your expertise and your time to enable this series of works to manifest materially. And thank you, everybody, for being here. Really appreciate your questions and your time. I hope that whatever that we had shared had been of interest. I'm really glad that we had this that we had this talk. So thank you so much.
Right. May I just share 3 quick thoughts in kind of a summary, and as well as perhaps a look into the future. The first thought I wanted to share was, as we started off talking about the different languages of DNA, the language of of the hexagonals, the hanacaraka, Javanese scripts, and so on. We are actually talking about something that is as old as man's conversation itself. The Rosetta Stone, which sits in the British Museum in London, was instrumental in being able to link 3 different languages together: the hieroglyphics of Egypt, ancient Egyptian itself, as well as ancient Greek. Today we have far more languages in our hands, and I don't mean computer languages alone, but that does include computer languages. But the more fundamentally is the language of thought. And while we evolve different languages to represent, different ideas, I think that linking these languages together, always having a common translation is important. And I think these relationships between artists and scientists, between doctors and engineers, and so on, needs to be propagated, especially in this very polarizing world that we find ourselves in today. A second thought I'd like to share, I mentioned computer languages, and if we were to look forwards, I think we can't ignore the elephant in the room, and that is artificial intelligence, machine learning, very hyper advanced technologies that can almost replace humans, or the threat, of course, is that it may replace humans indeed. And this is where the artist, I think, and the ethicist, the humanist, has a very, really important role to play in society moving forwards. And I'm talking about a very pragmatic role here. Technology has its pace on its own. But and we've seen that the adoption of technology can sometimes be very challenging. mRNA vaccines today, for instance, one of the newest vaccines we have. The vaccine for COVID has been developed in under a year, breakneck speed, world record speed. But yet we face some challenges in the adoption of these. There are anti-vaxxers and skeptics who may have valid reasons for what they think. And here is where I think this is a community effort, this is a human effort. We need to come together and address these issues. And the same will be true of computing and AI. Whether AI can replace an artist or a composer, I think, or a poet for that matter, remains to be seen. But this is where science and arts evermore need to be scientists and artists really need to be talking together very closely. And finally, I want to enlarge that thought beyond just science and arts to a larger language. And here I bring in this book by the director of National Institutes of Health, a notable geneticist himself, Francis Collins. And in a book where he describes the Human Genome Project, he describes it as that it's more than just DNA. It's more than just the DNA of humans. It's indeed even more than the language of the DNA of all living things. And here he tries to cross the even larger chasm between science and philosophy and morality. And I guess at the end of it all, I think that is what we are. We are more than a bag of genes. We are more than the sum of our creative works, be it artistic works or scientific works. We are, in a short, human. And I think that is the inspiration that I take with me, that I can tell, therefore, our work will never end. Thanks a lot.
Thank you. Thank you so much for those wise words and sharing, Dr. Yap. And indeed, why this entire project has started is because after my visit to the MIT Media Lab, which was also my first exposure to this very intense exposure into how art and science and even community, right, come together to address the complexities that we face, where neither art nor science can do without each other. So this is a first attempt in gathering some of the artists, such as Boedi, and last week we had Guang Yi. And in the following weeks, we would be having Tiffany and also Tuan Mami from Vietnam to share with us some of the practices and some of the ongoing inquiries. By no means complete, by no means exhaustive, but really probing, right? Probing into these ambiguous and these ambivalent lines between art and science and life and morality, and what kinds of future that we want to all imagine together. Yeah. So on that note, I think we will have to end today's session. Thank you so much for your kind attention and your time, and I hope to see you in the next sessions. Yeah. So have a good weekend and goodbye. Thank you.
Thank you, everybody.
Thank you.
Can we actually take a group photo before everybody leaves? Just because this is a project sponsored by the Ministry of Culture, and we want to take a record of this actually happening. So if everybody would be kind enough to reveal their faces, we could take a quick group picture before we end. And I put the links to our website, Instagram, and Facebook page on the notes if you want to continue following our event. I'm just going to wait a little bit for everybody to turn on their screen so I can take a screenshot. Oh, and everybody can say hi to the other artists.
Hello.
And everybody
Thank
can
you,
it's okay now.
Boedi, very much. Thank
All
you,
right.
Dr. Eric. It's very interesting.
Well, I took some photos. And everybody is welcome to chat with whoever now. We can just kind of talk. And if everybody wants to leave, they can leave now. Thank you, everybody, for coming. And they're going to just slowly leave. How are you? How are you guys?
Yeah, it was a good session. No, it was like a
Very interesting.
bit over time, but I think it was wonderful, Boedi. Thank you for your presentation. It was really clear.
I learned a lot.
Okay, so okay, you don't think that I was being too long-winded?
No, I think to have to kind of clarify your project and shared it in that 40 minutes was pretty amazing.
Okay.
Mm.
Cool.
Yeah, I think your work, the explanation really helps to understand your work.
Mm.
Okay.
Even though it's still like for me, it's still exploding my mind. It's very complexity, very deep.
Oh, I forgot to mention Henry Tang. Oh dear, Henry
thanks.
Tang, are you
Henry.
there?
Henry, are you here?
Henry, oh dear, I
Henry,
left out
open your
Henry.
camera.
Henry has fled.
His photo is still here.
Boedi and Dr. Eric, I'm a little bit curious about sort of I'm very bad at scientific, but I was thinking of I was wondering about how our future after the coronavirus vaccination. It probably it will change our and DNA, right? So, do you have any mind on researching or doing any work about it?
Eric, I think this is for you.
Well, thanks for your question. If you're referring to the question, if you're asking whether the vaccination dilutes or changes our DNA, and you're probably referring to the latest mRNA vaccines, the quick answer is no, it doesn't. And the reason we know that is that we have actually seen lots of instances in nature where our DNA a piece of DNA gets into our own genomes. You may be surprised to know that you and I, each one of us, are carrying DNA from a bacterium. And in fact, it's so critical that if you remove that that bacterial DNA from our cells, we'll all be dead. Yeah, and that bacterial DNA is actually called mitochondrial DNA, the one that I mentioned earlier, passed from mother to child. And yeah, so actually in nature, these things have happened before. DNA enters our genome. But here's the difference. That takes hundreds of millions of years to happen, or indeed billions of years. So in short, for the vaccine, in that 1 year, or even in the space of our whole human lifetime of say 70, 100 years, it's not likely to, it won't happen. So that's why I've taken my vaccine.
Thank you very much. It's very helpful.
Welcome.
Is there any other questions or feedback? That anyone wants to share? Yes. No.
Hi, Lorenana.
Okay, so maybe, huh?
No, I think that was really good. Good turnout as well, considering the diversity, the challenging topic that we have.
Yeah.
Yeah, but anyway, this is recorded, and I think it will go online, no? Yeah, so there will be more, post live audience.
Right.
Yes.
Okay.
Thank you, guys.
All
All right. Thank you so much.
Bye.
Thank
Have a
you,
good weekend,
everybody.
everybody.
Bye-bye.
Bye. See you.